Okay, just probably a couple more seconds here, waiting for people to come back in. But Craig, whenever you're ready, I say you can take it away.
Great. Will do. I hope folks are back. Just reminds me of teaching all spring with my university students, right? I've got no mug shots here, hardly at all, no live video feeds. I kind of speak into the abyss. So if you're out there, maybe you could like do a little icon with a thumbs up or something like that, just to see who might be among us in real time. Good. We got Brett and Rob and Josh, Justin. Okay, good, nice. Sounds good. I saw Olivia. She waved. Yeah, she's got her video feed on. That's great. And super. Hey, I appreciate that. So yeah, I'm Craig, and Keith is on video there too. You can see him over there. He's in my upper right-hand corner of this giant Hollywood Squares thing or Brady Bunch thing, whatever Zoom is these days. So I have a request too. I would hope that, well, first of all, I missed the first part of the meeting today. Our daughter gets married on Saturday. And so if you might imagine, I've got a few things going on, little details to tend to. And so sorry I missed out on the earlier talks. It sounded like you got a full tour of the Poor Farm, kind of a live thing. So hopefully that went pretty well. Our plan for the day is to show, so I've got a video. So I made a video out at the Poor Farm, really specifically the things that Keith and I have been interested in studying. I don't think would be too much overlap between, what I've got going on and what you've heard already. But, if there's a little overlap, that's okay too. And so that's about 15 minutes long. And then Keith is going to take it away for a while and give you some more details about what the Geological Survey has been doing at the site. And then I've got 3 slides at the end if you guys are really hungry to learn more about soil microbiology and the nitrogen cycle from a high level, right? And I just assume this be as interactive as it can be. And so I was really happy to join in late to see all these questions coming in and so on and so forth. And then along those lines with the chat, I don't know everybody here. I know a few names, but while the video is playing, or you can even start now, I can see all your names here, but if you could say, what you do or whatever, why you came to this meeting, so on and so forth, throw that in the chat and I'll kind of read that as the video is playing. Of course, I've already seen it, so I don't need to pay attention maybe as much as you might, but if that seems like a fair kind of a layout for the session, we'll move forward with that. Does that sound good, Olivia and Steve and others? Thumbs up. All right, good. And candidly, if you don't like that, I'm not sure what else I would do. That's what I came prepared to do. So hopefully it'll be all right. All right, so let's see if I can show the video. It was nominated for an Oscar in the short clip in the short film category. Just so you know, I'm joking, of course, but it's relatively well done for a hack like me. Go ahead, Steve, jump in.
Make sure when you go to share that you check those 2 boxes down at the bottom of the share window. And I'm sure you're familiar. I just wanted to throw that out there.
I did. No, I appreciate the reminder. I did learn that lesson a while back while teaching. My students had to teach me though, so that's good.
Right, thanks.
I know, I appreciate the reminder. And so with any luck, are you seeing just kind of a dark screen right now?
Yep, looks to be working.
There we go. All right, here we go. Hello, this is Professor Craig Just from the University of Iowa College of Engineering, and I'll be sharing information related to our EPA-funded project titled Connecting Rural and Peri-Urban Farmers to Demonstrate and Disseminate Innovative Nutrient and Sediment Reduction Practices. This particular program is funded by the Gulf of Mexico program, with the idea of trying to make improvements in the Gulf of Mexico by practices that we do in watersheds upstream of that location. What we're doing here in Iowa for this particular project has a total cost of a little over $1 million. And of that, about $350,000 will be dedicated to the installation of best management practices at various places in Clear Creek watershed, as it leverages practices that are being built with funding from the Iowa Watershed Approach. And then also we'll be doing some work down here just outside of the Clear Creek watershed at a place called the Johnson County Historic Poor Farm. What we're doing for this project overall is we want to demonstrate and disseminate real results from innovative nutrient and sediment reduction practices implemented in areas kind of where rural and peri-urban folks, where they meet, right? So really where, I should say where urban folks meet in these peri-urban areas. And the Johnson County Historic Poor Farm is one of those areas. We want to engage rural landowners, small communities, and metropolitan areas in this project. And we want to get some conversations going, farmer to farmer and farmer to consumer, so that then we can better implement some of these best management practices and improve water quality throughout our watersheds here in Iowa.
Welcome to the historic Johnson County Poor Farm.
Our activities that we plan for the Johnson County Historic Poor Farm are governed by the master plan that was adopted a few years ago. To orient yourself, if you do know Iowa City at all, here's Melrose Avenue as it crosses under Interstate 218. Coming through here, you can see the outline of the property. It's roughly 165 acres, give or take. Goes around this area. Here's the main entry point where you just saw the entry sign that leads in, and then here's a big red barn that was in the background there initially. Some of the practices that you'll see here, there'll be a met station right over here. As we kind of move down through this area in the site plan, you'll see these growing operations, but you'll also see a water control structure that will control the elevation of the shallow groundwater as it's impacted by the tile drainage that's in the area. Then kind of moving down through here, we're working on something called a grass waterway right here in this area. And then we'll also be installing a dam here to create a wetland then that will treat tile drainage effluent that comes into it in this area. Further to the south, eventually you can't see it very well in this image, but there is an alternative tile intake structure being built here that Keith Schilling can tell you more about. And then there's also some work that'll be done kind of roughly down in this area here in the immediate term with our EPA funding to potentially intercept the tile drainage outlet here so that then we can better treat that nitrate. Some changes have been made to this master plan over time, but you can see that they're hoping to have some trail access to bring the community into this area to see the different types of things that are going on. And if you haven't heard the latest about what's being planned for the Johnson County Historic Poor Farm site, please go to their website and ask them for more information. This is a hydrologic station installed in partnership with the Iowa Geological Survey and the Iowa Flood Center. This particular configuration measures rainfall, wind speed and direction, and also measures depth to shallow groundwater. The hydrologic station is shown here on the Iowa Flood Information System, otherwise known as IFIS. By clicking on the station itself, some general parameters are shown here on when the last report date was, what the groundwater temperature is, groundwater table depth, wind, and recent rain. You can get more detailed information here. And then you can also, for example, here's wind, and you can go back in time and see what's been going on. And then you can also see the wind direction as you kind of scroll through these data. Same thing for the well itself. You can get the groundwater or depth to groundwater levels at that particular location over time. This particular site does not have the soil moisture and soil temperature probes installed, but you could get that information at other locations. And then of course we're in a bit of a drought at the moment, and so there hasn't been any rain hardly at all at the site in the past week or so. Hopefully we'll get some soon. One of the objectives of this EPA Farmer to Farmer grant is to better understand and to have more conversations about the impact of tile drainage on our watersheds in Iowa. Here on the left is the drainage network that was installed at the Johnson County Historic Poor Farm a few years ago. For orientation, here's that big red barn on your way in. Here's the meteorological station, or the hydrologic station that I showed earlier. And there's tile about every 30 feet apart on these diagonals like this, that then are collected then by a kind of a central collection tile that then goes along here, goes all the way down and daylights into a swale that you can see over here in this picture, right on the right hand side. And the same thing happens on the west side of the property. Again, several of these diagonal tiles capturing the water, and then again moving it down through this central tile channel all the way down before again combining into that single PVC pipe shown in the picture on the right. One of the things that I want to point out here, since I will show you an image of that in a second, are these water control structures they've got here. So they've got them labeled. There's actually one way up here, but there's one here labeled number 2, one here labeled number 3, 4, 5, and 6. And these water control structures are what can be adjusted to alter the shallow groundwater in ways that can make nitrate processing, or nutrient processing in general, but certainly nitrogen processing, more powerful, and can have a wonderful impact on our water quality that leaves this tile drainage network. The idea behind a water level control structure, as illustrated by the NRCS as part of their drainage water management approach, you can show here that you can take drainage tile that has been put in the ground, you know, as much as 5 foot deep, certainly at least 4 foot deep. You can have that tile intersect this water level control structure that has a series of adjustable riser boards inside, and you can stack those up. Depending on how you stack those, they become, you know, a weir, and ultimately they control then the groundwater table on the other side of this structure. The water then moves over this weir and then can drain out into the drainage area as would normally be the case. And here is a water level control structure at the actual historic Poor Farm site. If we look at the target water levels with such a system, we might look at the growing season kind of after harvest, looking here in October, somewhere in that sort of a date, and seeing that at that particular time there's a minimum water table depth of inches below ground surface of about 6 inches. So we've got that groundwater perched pretty high, and you leave it there during the fallow period. Then when it comes time to plant, you pull all of those risers out of the water level control structure and drain the soil profile all the way down prior to planting, so you don't have any issues with equipment getting in and out of the fields. Then after planting, you can raise that water level back up and store it during the growing season, and you can get it perched up while the roots are relatively shallow. As the roots start to increase their depth, you can then slowly pull each of those risers out of that control structure and gradually lower that water table. Bank it all summer long, and then near harvest time, when you want to get the equipment in the fields again, you pull all of the risers up and out of there and drain all the water out. really the 1, 2, 3, 4 steps required for this. Again, number one, you remove the remove riser boards to drop water table levels about 10 days before planting. During the growing season, you stack those riser boards to raise the water table high enough to provide capillary water to the crop root zone. Third step is to remove the boards to lower the water table 10 days before harvest. And then last, after the harvest, you want to raise the water table even further near ground surface to hold nutrients in the soil. More specifically though too, while this soil profile is saturated with groundwater, it becomes anaerobic. It loses its oxygen. And the soil microbiology then can also convert nitrate to nitrogen gas when it's in that state. And that's how we end up treating our nitrogen issue here in Iowa.
This nitrate sensor uses ultraviolet light to measure the amount of nitrate in the water that's coming out of the drainage tile. The measurements can occur every 15 minutes, could be every hour, could be once a day. It's completely programmable, and it gives us a real-time view of what's going on in the groundwater that's being captured by the tile drainage network. The information from the sensor is wired into this control box. Inside there's a battery and a small computer and a cellular modem that can send the data to the Iowa Flood Center servers that push the data to the Iowa Water Quality Information System shown here. We can see a variety of nitrate sensors around the state, and if we zoom in to the one at the Johnson County Historic Poor Farm, right here, we can switch over here to satellite view, and you can see the Poor Farm site, and then we can click on the sensor and we can view the data as it's been being received over the last few days. We can look at an annual trace here and see that back here when we were first kind of getting going for the season, it was about 3.5 milligrams per liter as N, and then you can see it kind of doing its thing here over time as the water table moves. As the water table changes, the concentration of nitrate can change as well, and so those are one of the things that we want to be able to do, is to look at this concentration over time as a function of rainfall and groundwater saturation and so on and so forth. One of the things that we're interested in doing is looking at these areas like shown here, that in a heavily row cropped area would be a grass waterway, and whether or not we can intercept tile already in the field, put in water control structures like we've shown you here, and then use that soil profile to treat nitrogen. Thank you for listening.
All right, I think it's pretty self-evident on why that got nominated for so many awards. But you know, I enjoyed putting that together, and I didn't cringe too many times as I get to watch it back myself. So we'll have plenty of time for questions as the session moves on, but I think now it'd be best to throw to Keith and let Keith share a few more details on what's going on out there. And then before we do that, I just want to thank you too. I see several people drop some information in the chat box, and that was nice to read through during the video. I really appreciate that. So Keith, take it away.
All right, let's see if I can share this. Perhaps. You got it?
Got it.
Great. Yeah, so I'm going to follow up a little bit from Craig, talking more specifically about the practices that we're interested in monitoring out at the Poor Farm so far. I mean, there's more practices to come, including the wetland construction and alternative tile intake that I won't be talking about right now, but we'll highlight two of them. Activities will relate to what Craig was talking about with the controlled drainage and drainage water management. We've done some initial spatial characterization at the site and monitoring. Going to end with a new grass waterway design that we're contemplating out there. And in the future, of course, we'll have information to share on the wetland construction. We've got the monitoring in place actually to do before, during, and after construction. So it's kind of an interesting, unique kind of case where we can look at conditions, again, before, during, and after. So I think that's a unique perspective that we can bring. So like Craig was talking about, one of the things we are looking at is this idea of controlled drainage and drainage water management. Craig covered all this. But I will show a little bit of the data that we've got so far. You know, a drought is not a good time to do this. So we're all dressed up, we're ready to go. And we've got some initial early stuff when we had water, and now it's just been dry as a bone like it is across the state. So we're looking at implementing this. We've put all the boards in right now, and right now the water table is below all the tile networks. So there's not much we're gonna share. Again, Craig talked about this. This is the tile network more digitized on an aerial map. The green here are wells that we've installed. So we've got some monitoring wells we've installed in the drainage system itself. And so the idea is that we can monitor the shallow water table fluctuations as this controlled drainage is going on. So we're not going to be just looking at the export of water from the outlet. We're going to be looking at the entire behavior in terms of the groundwater system. So again, here are the wells across here. The drainage system extends all the way into these other fields. Those are the 4 wells. We did an initial test when it was wetter out, put all the boards in, backed the water up, again, as this kind of controlled weir. And within a 6-hour period, we were starting to see water table rise in some of the monitoring wells. So it indicated to us that this was a pretty, valid effort to try and monitor these conditions. So within the 6-hour period, we had several inches of water table rise. Of course, that was when we actually had water. Right now, again, I got all the boards in, or not seeing anything. This is the ongoing. These are all the transducers that we have out at the Poor Farm site. We can see some greater fluctuations in the well, kind of where the hydro monitoring station in places. Otherwise, these handful of wells here are the ones in the water table in the controlled drainage area. You kind of see that they're below 5 feet at this point, and they're just behaving naturally until we get water table rise. Some of the shallower ones are in the wetland area, or the low areas there that have naturally higher water tables. But like we're all dressed up, ready to go when we have the water and precip occur. I wanted to talk about a parallel site that we've been monitoring up at Kirkwood Community College in Cedar Rapids, because we're using a lot of these same techniques and extending some of our results to the Poor Farm site. This kind of gets at some of the characterization that we're doing. At Kirkwood in southern Cedar Rapids, you kind of see the map here. We were assisting with Kirkwood and the Iowa Soybean Association to do some monitoring associated with the strip trial test, where we looked at different nitrogen forms and placement methods in a field, agricultural field that they have in production. So Kirkwood generously allowed us to put monitoring wells in this field, kind of flush with the ground so they could still farm, and we were able to monitor conditions over a couple-year period. On the surface, you'd say, hey, there's just not a whole lot outside of this strip trial. It's relatively flat. You kind of see that from the topography, and you might not think there's much subsurface lithological variations in there. But instead, we found there was a major difference at the site that seemed to exert overwhelming control on what the groundwater conditions were. So we did this middle feature here. We walked the site with some hydrological survey geophysical equipment. This is other equipment we have, electrical resistivity, where we can image below the surface, upwards of several hundred feet. And we also have this passive seismic unit. If we want to measure, estimate the depth of bedrock in certain places, we can put this unit down and just get an idea where the bedrock surface is. But for the Kirkwood site and for the Poor Farm site, we walk the area with this electromagnetic induction tool. It's a geophysical unit that sends a electrical current down below the ground to about 6 meters, 18 feet. It passes through the ground, and then it's picked up by a receiver coil. And as you walk around, you can get the average ground conductivity at your spot as it sends that current down and picks it up in the receiver coil. So we did that at the Kirkwood site and saw that there was a major difference in lithology at the site, again, that you would not necessarily see from walking around, where it had half of it was in a fine textured glacial till, and the other half was underlain by a sandy outwash alluvium, with a very sharp boundary as we walk that entire site. Here's the lines would indicate the strip trial. And then what it turns out, we had these wells in both environments. We saw major differences in subsurface water quality that really dominated over the strip trial test. The groundwater within the fine textured glacial till had higher dissolved phosphorus, sulfate, and specific conductance. And in the sandy outwash alluvium, coarse textured, it had a deeper water table, and groundwater quality was higher, much higher in nitrate and dissolved oxygen. So we could see spatial variations in groundwater quality out here, due mainly to what the subsurface looked like. In terms of the water table, here was the differences between the water table and the coarse textured aquifer that drains a lot better. So it's just going to be a deeper water table compared to the fine textured till that retains the water more and kind of keeps the level higher and more responsive to rainfall inputs. So with that in mind, we had this example of how effective this technique was out in Kirkwood. We did this same EM survey at the Poor Farm site, and this is the results kind of overlaid on the air photo for the site. And you can see the differences in the colors between the cool and the warm colors, with the warm colors being that fine textured material, and the cooler colors being a little more uniform, kind of lush material. So we had definitely a more clay-rich, fine textured environment in the waterways. This is one waterway, and you come up and over a hill, and there's another waterway. So the idea being that we could essentially map the subsurface. If you'd wanted to figure this out without doing this geophysics, you would have had to, grid the entire site with boreholes to figure out how these patterns vary. So we're able to do that and helps understand the background conditions at the Poor Farm. Another thing we were looking at Kirkwood that we want to extend and test out at the Poor Farm is this idea of what water quality benefits some of the subsurface deposits in the waterway, grass waterways, might have on processing nitrate. We posed a question at Kirkwood, and we're going to assess in more detail at the Poor Farm, is that can these saturated waterways be roughly analogous to saturated buffers? So at Kirkwood, here's the college, and we installed monitoring wells in several different grass waterway complexes, and to look at the water quality conditions there. So if you look at water quality treatment in waterways, you see that there's typically, you know, the waterways occupy the low areas and therefore have received sediment deposition from erosion from the upland areas. And if you take a cross section through what these might look like, especially across the area of Iowa City, southern Iowa that we're in, you might see loess, this 5 unit overlying a glacial till. And in the waterways, you have a different kind of deposit there that's kind of the receive the erosion and deposition from former topsoils and erosion of the loess and so forth, and the winnowings of some of the coarser materials. So it tends to be finer textured in the waterways. If you took, we analyzed this at the Neil Smith Refuge in Jasper County, and what we found there is that the water tables in these drainage ways, let's look at the bottom graph, the water table in the waterway stays relatively flat, very, very saturated, stays that way all the time. It's the low spot in the landscape. But the water table in the upland areas where this other monitoring wells would be out here vary a lot. Okay, it might rise and fall upwards of, you know, 3, 4, or 5 meters. Okay, so major fluctuations in the uplands, a very constant saturated environment in the waterways. And if you look in terms of the groundwater chemistry, that this catchment scale, let's look at dissolved oxygen concentrations in the waterway are typically very low. And because of that and some of the carbon that's available, we typically get a lot of denitrification occurring in the waterway. And the concentrations of nitrate in the upland fields might be 10 or 15 milligrams per liter. And in this case, the waterway was less than 1. So you see this great environment conducive for groundwater processing, and it's really has a similar kind of characteristics as you might think of a riparian buffer. So we went out and installed some monitoring wells, both to investigate this and those other practices. The Iowa Geological Survey has our own Giddings drill rig. Here's Matthew Streeter, who's a licensed well driller. Should anybody be interested in working with us on projects. So he's collecting a soil core there. At Kirkwood, looking at this top graph, it's exactly what we've seen before, which is the water table in the waterways is very, very high. So, you know, less than a meter below ground surface, it fluctuates very responsibly, and the water table tends to be really high, and they're just saturated. If you look at the the water chemistry, again, it's exactly the same picture as we've seen before, only here it's all over the place at Kirkwood, where water nitrate concentrations in the waterway, in this case, were less than 2, and in the surrounding ag fields, they're upwards of 10 to 20 plus milligrams per liter. Did not see significant differences in phosphorus, chloride, or sulfate, although we did see some higher values of phosphorus in some of the saturated alluvium. But this is really interesting, you know, like, can we use this information to say, just like a saturated buffer, can we come up with something that might be like that in terms of a saturated waterway? Most of the grass waterways in Iowa are in fact tile drained. If you see them being installed, there's typically a drain on both sides of the waterway that are taking that drainage water down to the outlet and discharging into a stream. Is there some possibilities of connecting these to a central tile and maybe encourage some nutrient processing of that tile water through this environment that's very conducive for that sort of treatment? So this is a practice that we're looking to investigate at the Poor Farm, because you know, that's kind of what this idea of this EPA project is all about, is demonstrating some innovative practices. And, you know, the opportunity is there. So there's a couple different waterways that we're looking at working at. Let me back up for a second. So this is Hoosier Creek Waterway, Hoosier Creek Watershed. Here's Kirkwood in here. Okay. What we showed in this is when we did the ACPF tool for that entire waterway or watershed. All of these red areas are potential grass waterways that are likely tile drained. So if you see this idea that this practice that we can perhaps work with the saturated conditions in a waterway to get some processing. That's not an isolated occurrence. They're everywhere, especially in eastern and southern Iowa. So lastly, here are we've two waterways we're interested in investigating out at the Poor Farm. We've installed some. We first selected this one on the right here. It tends to be a little steeper than what we're interested in. Both. Then we've just recently installed some more wells in this one. This is the waterway that's going to feed the wetland. In both cases, again, we have the right biogeochemical conditions where this is occurring, and we will configure these at a later date to try and test this idea of nitrate processing. And that's what I have. I'll turn it back over to Craig.
Great. Yeah, that was awesome. I even knew what you were going to say, and it was awesome again. So I think now it makes the most sense to pause for questions and comments from the group. Be happy to add some information about soil microbiology if folks are interested, but I think we should pause here and see where we're at. So I'll open it up. Who's got a question, comments? I guess I can monitor the chat box as well if they dump there, or otherwise just unmute and let us hear your voice. Any at all?
I've got one, I guess. Does drought conditions like we've been dealing with have any long-term impacts on the drainage management system that you guys have installed? Like, does it need to be wet at any point during the year to not have future maintenance issues or anything like that?
Well, I wouldn't. I don't anticipate that. I think it's going to work as long as we got water. So again, once that water table dropped below the tile depth, we're just pretty much in a waiting mode at this point. We'll see though, if we get the water table to come back up. I'm anxious to see what sort of management we can do. Craig mentioned, this is a slight off tangent, but the nitrotax unit that we have at the outlet of the tiles, well, we also have one back in our lab. So we're able to collect a lot of water samples during this process and get, essentially a free nitrate test very regularly. So should we get the water flowing again and start working on backing up some water into the system, we'll be able to test the wells, the tile boxes themselves, and also the outlet, kind of see how all this kind of might, reflect some of the management.
Any others?
Yes, how would this practice be different than a wetland as far as denitrification practices, or how would you, I guess, market it to a producer?
Well, which one? Are you thinking the controlled drainage or that waterway practice?
The waterway with the retention.
Yeah. Well, you wouldn't have a wetland on your property. So it's like in some ways it's kind of a buried treatment system. I would say, it's got a lot of potential, although we're still in the very early stages. And some of the challenges that we have here are some of this alluvium in the waterways is very fine textured and it has a very low ability to have groundwater flowing through it. So we're really going to have to analyze the right particle size distribution and hydraulic connectivity of these materials to say when can it be most effective? So there's always a balance between, you know, how much water it can treat versus, you know, backing up water and impacting the drainage. So that's one thing we're going to have to investigate. But you know, I think there's a lot of room for growth here. We'll see how this turns out.
Yeah, that was going to kind of be my follow-up. So, you know, if the testing indicates that the practice is a good option in areas where the soils fit, you know, the kind of work you're intending the practice to do, is there, you know, a break point in some farm areas where maybe amending soils to an existing waterway would be worthwhile? You know, maybe this is a new edge of field practice kind of a thing where if the native soils there aren't always suitable, it could be a good area to, like a saturated buffer, build it in place and amend some of those soils. So interesting questions, and we'll be watching that one too, that research. So.
Right. Yep. It's exactly right. I mean, I don't think this is a practice that will be a good fit for everywhere. I'm thinking, you know, we'll see where it goes in the poor farm. I'm thinking sometimes it might take a little bit higher hydraulic conductivity, a little more sand content, to allow enough water to flow through it and be processed to make it worthwhile. You know, so I guarantee you that every one of these waterways is doing exactly the same biogeochemistry. Now it's a matter of can it see enough water to be worth the effort, you know? And so there's going to be a line there where if you look at the texture of the materials or have some way of assessing how well it can transmit water, you're going to say, does it fit or not? You know, and if it fits, I think could be a practice. If not, you might be looking at, you know, edge of tile practices or something like that rather than an infield practice.
Think sediment transport is going to be an issue?
I don't anticipate that unless we find some way to get surface inlets into it. I don't really see that unless, you know, they're attached to the system somewhere. So I think it's gonna, like I mentioned, it will work everywhere in terms of the biogeochemistry. I just feel like I've seen enough of these now. It's like that's the easy part. It's just whether we can get enough flow through them to be worthwhile.
Anybody else? The threat's real. I'm going to start going into soil microbiology here pretty soon.
I
So
can probably throw another one in there. So we talked earlier when Justin and Jason were on about the blind inlet practice. Outside of maybe some potential information that you would gather doing your you know, the research you're doing on the poor farm, are you aware of any other areas where they're doing any research on blind inlets in the amount of nutrient treatment they can do?
Yeah, actually, we're just finishing up a 2-year NRCS conservation innovation grant on blind inlets. So we did monitoring at 3 different blind inlet sites, 1 in Clear Creek and 2 down in kind of southeast Iowa. You know, COVID kind of gave us you know, we pretty much took a year off with the COVID. We picked it up again this spring, and we're still kind of analyzing the data. But the amount of processing there of water and nutrients is really kind of impressive. So I think it's going to be a good story. None of these are very accessible to go out and see. You know, they tend to be a real, you know, hard to get to and so forth. So what's the advantage of trying to implement this practice at the poor farm? I think we could do it in such a way to highlight what's going on and maybe have a good educational tool to show. But we looked at the nitrate process. This is an ATI that had not just the inlet, the different inlet type with maybe rock and so forth, but also we've also put a layer of wood chips in there. So now we have some sediment particulate, treatment, and also the nitrate treatment from the flowing some of that water through the wood chips. So it's all really promising, and we're still analyzing it, quantifying the data.
All right, I guess it's time for soil microbiology lesson.
It's going to be a good one now. All right, it's going to be good. And Keith, I think you said you might have to go. So if you break away, I won't take it personally, right? I knew you might have to go. So, all right, I'll share my screen real quick and just provide a little extra detail here about some of the consequences of doing that. You guys seeing this slide now? My little laser pointer? I can't see any of you, by the way, so I need a verbal.
Yep, it looks good.
Thank you very much. So this is the target water level settings, example plan that I showed in the video, right? And so when the water table is high and, the soil profile is, really saturated down in here, it starts to become, anaerobic. It loses its oxygen, of course, up here, in the non-saturated zone. There's some oxygen transfer that can get in there from the atmosphere and can keep things much more aerobic. Well, there are consequences to that when you try to understand the soil microbiology in play that's going to then do this nutrient processing that we're talking about. And so you can imagine this red line as it's going up and down, right? Red line going up and down here through the season, or however the case may be, being this line here, the line in between this green area, which is oxic, and this kind of this lighter tan area, which would be anoxic or anaerobic, right? And so in this case, it's a very detailed nitrogen cycle. I don't want you to get too overwhelmed by it. In a nutshell, ammonia or ammonium can be nitrified by a variety of organisms. In this case, I'm showing ammonia oxidizing bacteria and AOB and ammonia oxidizing archaea, AOAs. And so they convert this thing to nitrite, and then nitrite by a nitrite oxidizing bacteria can get fully oxidized to nitrate, right? And so this is the big water quality issue we talk about here in Iowa is nitrate. It's very mobile, doesn't absorb the soil. In really fast moving waters, it gets transported with it without really much processing. And that really goes to Keith's point that you got to have a little bit of residence time in these anaerobic zones to get this nitrite or nitrate converted back to nitrogen gas. So let's talk about what that would look like. You can then go back and under anaerobic conditions, you can convert back to nitrite, and you can cascade down this classic chain of denitrification, right? So this is the nitrogen cycle I was taught 20 years ago, just this part, where you go to nitric oxide, nitrous oxide, and then to nitrogen gas. An engineer's dream. If you can make nitrogen gas, it bubbles out. We use this exact same process all the time in wastewater treatment. That's how we remove nitrogen and try to not discharge it to our rivers and streams after we treat all your flushes. Same thing is going on in the soil profile here as well. And so this is classic denitrification. And this classic pathway is why we always want to put organic carbon in these blind inlets or in these grass waterways. These organisms are dependent on yummy organic carbon to do their work. So it's got to be anaerobic, and there's got to be plenty of yummy carbon. And so that's why we, emphasize the use of like a wood chip bioreactor. You need those wood chips to just supply the carbon, such that these organisms under anaerobic conditions can make this nitrogen gas. There are some other pathways that we study. I won't dwell on Anamox, but it's anaerobic ammonia oxidation, which is ironic to think you can oxidize something in an anaerobic condition, but you can. And we're using this in wastewater treatment a lot, and Anamox organisms are around in the environment too, and they've got some benefits. But I want to focus just for a moment on this N-DAMO, right here, and that is nitrite-dependent anaerobic methane oxidation. Again, it feels like a bit of an oxymoron that you can have an anaerobic oxidation. It doesn't seem like something that you would think much about. But there's some really advantages to this, and one of the things we want to study while we're, engineering these water tables in these controlled drainage systems is, can we preferentially shunt this nitrate reduction through this process. The reason why you'd want to do that is it consumes methane, which is a greenhouse gas. It'd be great to, and CO2 is a greenhouse gas as well, but methane is 23 times more potent. And so you just assume have the soil convert that methane for you into CO2 because you'll get a climate change benefit from that. The other thing about this process is that it skips nitrous oxide formation, another greenhouse gas that's 300 times more potent than CO2. And so if you can figure out how to engineer these systems in a way that even partially favors this pathway over the classic denitrification pathway, then you not only get your water quality benefit, but you get some greenhouse gas benefits. And I think that's a pretty powerful thing. And again, we're doing these same things in wastewater treatment as well in much more controlled settings, but I think it'd be great to be able to apply that in natural environmental systems. The way that we do this is we go hunting for DNA. And so we can determine sediment microbial community composition by getting a sample, right? So we go out there and in some of these murky areas in the pore farm, or we also have extracted DNA from the cores that the Iowa Geological Survey has pulled out and actually measured the DNA at various depths along the way to try to find that niche, right? To try to find that zone between oxic conditions and anoxic conditions, because that's where we think these particular organisms are going to thrive. We can extract that DNA. We can chop that DNA up and look for a very specific part of the DNA, which is called the 16S region. We can amplify that DNA. We can align that DNA and create something called OTUs, and we can compare these OTUs to a database. And then we can identify which organisms are there, and we can kind of get a family tree, right? So we can get a phylogeny. We can figure out, who's there, and then what is the relative abundance of all of those organisms in that community. And so we can follow that change over time if we want to see how the community changes over time. That's a pretty powerful thing to do. A slightly more, well, not slightly, quite a bit more sophisticated approach, it's more expensive and takes a lot more time, is you can take that same sample and again extract the DNA, but then you do what's called shotgun metagenomics, and that's how the human genome has been sequenced, right? You break up all the DNA into a bunch of tiny pieces, you amplify it all, and then you kind of piece it back together computationally, right? And so then here you've got some small pieces, you stitch it back together in a big computer. It takes a little bit of processing time. There'd be millions and millions and millions of DNA reads in this case. Then you run it through a few databases, and then you can start to understand the abundance of various gene pathways. What this enables us to do is to figure out community function, right? So function is really important here, meaning are the genes there to code for the different nitrogen processes that we're interested in? And can we track those gene abundances over time? Again, as an indicator of our success or lack thereof, of our ability to engineer these systems to kind of go down the favorable pathways that we might want. And so those are some things that we're doing out at that site as well with my PhD student, Emily Schmitz, who is awesome, by the way. So I'll stop sharing. And with that 3-minute microbial community analysis talk, I'll ask if there's any more questions out there. Questions or comments?
I'm very curious now. You've made me want to investigate this more. So that's a win, I think, as far as I'm concerned.
Good.
Yeah, I rethinking the denitrification pathway. Yes, there's a lot of learning that kind of goes out the window for us who've kind of been in the field for long enough to have it ingrained. So
yeah, and it's still going to go down, predominantly down that major pathway. I would say it's extremely exciting for wastewater treatment folks like me, because the other pathways require a lot less energy, often a lot less oxygen. Oxygen equals energy and money. And so as we try to, you know, lower the carbon intensity of our wastewater treatment processes that we can do in tanks, we can control it, we got sensors, you know, I think there's a huge opportunity to make some differences. In these natural systems or these, you know, agriculturally influenced systems, you know, I think our ability to kind of dial that in is going to be pretty low, right? That groundwater table is going to be moving up and down. That oxic zone is going to be changing. The bacteria don't like it if you mess up their house every 3 months and move the water table someplace else. I think that goes to the question of, you know, during a dry year, are there impacts on how this will work? Well, your biology kind of dies off a little bit there, right? I mean, just like if you over apply, you know, herbicides, pesticides, so on and so forth, your soil health goes way down, you lose all those benefits. Well, in a drought year, these organisms are going to take a little while to recover once it gets wet again. They got to grow up their populations, and they grow pretty fast, some of them, but some of these niche organisms grow pretty slow. And so, you know, if you kill them all off during a drought, you just may not get those benefits in a year like that. And then same thing, if you're moving that water table up and down all the time and they can't really establish their niche, then again, you might be defeating kind of the purpose there too. So a lot harder to do it at a place like the port farm. But we're going to try. Okay. Very cool. I mean, I would be outside. I hope you're all on cell phones, candidly, out roaming the outdoors. I don't know. I feel for you guys. This could have been at the port farm. I'm sad it wasn't, but anyway, who knew, right? I mean, back in the planning stages, it had to be this way. We just didn't know with enough advanced time to meet out there, but I'll be happy to host you out there sometime. Anybody that wants to stop by, let me know.
Yeah, I think you know, I'm certainly interested just individually, but I think there's a lot of opportunities for the chapter going forward to plan a little bit more of these relatively soon in person again. So we'll be happy to do that. We just felt like this was the safer option for the moment, as everyone can probably imagine. So
yep, that's the right thing to do. All right, anything else, Olivia? I don't want to keep anybody if we don't have any questions. So
Yeah, I think that's if everyone doesn't have any more questions or comments for Craig here. I'd like to thank you, Craig. I think that's incredibly interesting that we can change the nitrogen